Okay, so welcome to this video. In this video what we're going to do is we're going to discuss a very important signaling pathway that is downstream mainly of receptor tyrosine kinases. Okay, right, and this is the RAS, RAF, MEK, ERK pathway. Okay, and sometimes it will just be called the MAP kinase ERK pathway. There are absolutely loads of different names for this pathway, and there's absolutely loads of different names for all of the components of this pathway. Okay, so ERK, the enzyme ERK, has other names as well. It also is called the MAP kinase, MEK, so sometimes you'll see it referred to as MEK rather than ERK, but this is what we're going to call it anyway. Okay, and this describes pretty much a cascade here. RAS activates RAF, activates MEK, activates ERK, as we'll see. Okay, right, so where to begin then? What is the structure for this video going to be? Well, we're going to start with receptor tyrosine kinases. We are going to see the basic structure of receptor tyrosine kinases, at least of most receptor tyrosine kinases. We'll then look at how they're going to be activated by ligand binding to them, okay, and then how they're going to activate this downstream pathway. So, then let's start off with a receptor tyrosine kinase. Now, for short, receptor tyrosine kinases are abbreviated to RTKs. Okay, so this stands for receptor, that's the R, okay? Then the T is for tyrosine, and then the K is for kinase. Okay, so these are receptors which have inbuilt tyrosine kinase enzymes within their cytoplasmic tails, basically. Okay, now, this pathway is going to be very general. It's going to be activated by a huge number of different receptor tyrosine kinase enzymes. However, we're going to take a nice example, basically, so that we've got something to work with, rather than keeping it utterly general. But the example of a receptor tyrosine kinase that we're going to take has a very sort of generic receptor tyrosine kinase structure, so we could be talking about many others. So there are around 58 receptor tyrosine kinases in humans, okay, and they're grouped into about 20 different families. The one we're going to use as an example is the epidermal growth factor receptor, okay, often abbreviated to the EGFR. So this stands for the epidermal growth factor receptor. So EGF stands for the epidermal growth factor. So it tells you exactly what the ligand for this receptor is. And then the R is for receptor. Okay, so the epidermal growth factor receptor is an example of a receptor tyrosine kinase. Now, it also has a bunch of other names, so I'll just tell you about the other names for the epidermal growth factor receptor. It's also called ERB1, so you might see it referred to as that, and it's also called human epidermal growth factor receptor 1, or HER1. So HER1 is short for human, that's the H, and then the E is for epidermal growth factor, which I'll just abbreviate to EGF, and then the R is for receptor. And luckily the number nicely adds up, nicely matches the number that we have on B1 as well. Okay, right, so these are the three different names for the same receptor tyrosine kinase, which is a receptor for epidermal growth factor. Okay, right, so this is the archetypal example of a receptor tyrosine kinase that we're going to illustrate this pathway with. Okay, so let's see the structure then of the generic receptor tyrosine kinase, which is the structure that epidermal growth factor receptor abides by. Okay, so we have the amino terminus extracellularly. Okay, and I suppose I should probably draw the cell membrane first. That's a more natural place to start. So let's say these two lines here represent the outer leaflet of the phospholipid bilayer, which means the outer layer of phospholipids that faces out to the extracellular fluid. So this over here is the extracellular fluid, or ECF for short. Okay, and then we've also got the inner leaflet of the phospholipid bilayer, which is the layer of phospholipids which faces into the cytoplasm, which is down here. Okay, now receptor tyrosine kinases are all integral membrane proteins, so they all sit within the membrane. Now their amino terminus is going to be extracellularly because they are all single polypeptides. They're not complexes of multiple polypeptides. They are a single polypeptide. Okay, so the amino terminus is extracellularly. And then a very important domain that you have extracellularly is then your ligand binding domain. Okay, the portion that will bind to the ligand molecule, in this case epidermal growth factor. Okay, now this is the portion that is highly variable between the different receptor tyrosine kinases, clearly because they all have to bind different ligands, okay? So the ligand binding domain, and for short, the ligand binding domain is often abbreviated to the LBD. L for ligand, B for binding, and D for domain. So I'll colour in this ligand binding domain here in orange. Okay, there we go. Then what you have is a membrane spanning alpha helix after this ligand binding domain. So now what we're going to have is an alpha helix that's going to straddle the membrane. Okay, so remember, an alpha helix is a spring-like structure. So if I draw a bigger picture of this, it will look like this. Okay, so the polypeptide folds up into this spring-like shape, and you'll have a lot of hydrophobic residues throughout this alpha helix, and these hydrophobic residues will interact favorably with the hydrophobic core of the phospholipid bilayer here. Okay, then the protein gets through the membrane, and then on the cytoplasmic side you then have another important domain, which will be the tyrosine kinase domain. So this is the portion after which the entire family of receptor tyrosine kinases is named. It's the tyrosine kinase domain. It's the domain which actually has enzymic activity as a tyrosine kinase. It can phosphorylate tyrosine residues on proteins, which I'll discuss more in a moment. Okay, so we'll colour in the tyrosine kinase domain here in blue. So this is the tyrosine kinase domain. Okay, I think we'll also colour in the membrane spanning alpha helix in red here. Okay, like so. And then we've got the carboxylic acid terminus of our polypeptide on the intracellular side, facing into the cytoplasm down here. Okay, so that's the basic structure then of a receptor tyrosine kinase. Most receptor tyrosine kinases look like this. Okay, they have a single membrane spanning alpha helix. That's a pretty unanimous feature. Okay, right. So what we now want to discuss is what a tyrosine kinase enzyme actually is. Okay, so a tyrosine kinase enzyme is just any enzyme that's capable of phosphorylating tyrosine residues within proteins. Okay, and what these receptor tyrosine kinases are is they're just receptors which have an inbuilt tyrosine kinase, okay? So there are many examples of other tyrosine kinases, some of which we'll come across later, which are just free in the cytoplasm, okay? They're not attached to receptors. This is special because this is actually a tyrosine kinase enzyme that's part of a receptor, basically. It's in the same polypeptide as another portion that makes up a receptor, basically. Okay, so let me just describe phosphorylation of tyrosine residues then. So let's start by discussing the structure of a tyrosine residue. So here's the amino group, and I'm drawing a residue. So what that means is I'm drawing the amino acid as though it's within a polypeptide, rather than just drawing an amino acid as though it's free. Okay, so here's the amino group, and I've drawn it as though it's bound to the carboxylic acid group of the amino acid before it. And then we've got an alpha carbon here with a hydrogen coming off, and then a carboxylic acid group here, which will be bound to the amino group of the next amino acid along. And now the R group of the tyrosine residue consists of a methylene group, like so, which has then got a benzene ring coming off it. Okay, so here's a benzene ring. And then off the fourth carbon of the benzene ring, you've then got an alcohol group. So this is a tyrosine residue. Okay, right. Now, what you can do then is you can phosphorylate tyrosine residues, which means you can add phosphate groups onto these tyrosine residues. So let me show you the structure of a phosphate group. So a phosphate group consists of a phosphorus atom double bound to an oxygen atom. Then the phosphorus atom has two alcohol groups coming off either side. and then it also has a single bond to an oxygen atom, which then needs to acquire a further electron via ionic means, and therefore has a negative charge. Okay, so this is the structure then of a phosphate group. Now, how then do phosphate groups attach onto alcohol groups? Well, there's a clue. If I circle this portion in purple here, what does that look like? Look at that group within the phosphate group. This looks like a carboxylic acid group. Okay, in fact, if that phosphorus atom was a carbon atom, this would be a carboxylic acid group. Now, carboxylic acid groups can interact with alcohol groups via ester links, and in fact, phosphate groups can interact with alcohol groups in a very similar way to the way that a carboxylic acid group would. Okay, so what's going to happen is the alcohol group is going to come off the phosphorus atom. The hydrogen is going to come off the alcohol group here. Those two things are going to combine together to make water. Okay? And then the oxygen atom of the alcohol group is going to bind to the phosphorus atom of the phosphate group. Now this link, which is very analogous to an ester link, this is called a phosphoester link. Okay, so you can attach phosphate groups onto tyrosine residues via these links that are very similar to ester links, and which are called phosphoester links. Okay, so I'll colour in this phosphoester link in blue here. Now, once you have attached a phosphate group onto a tyrosine residue in this way, the resulting structure, instead of being called a tyrosine residue, is instead called a phosphotyrosine residue. Okay, right. Now, what we want to discuss also is the fact that although I've shown the addition of a phosphate group onto this tyrosine residue as adding on an inorganic phosphate like so, tyrosine kinase enzymes do not just take an inorganic phosphate group like this that is free within the cytoplasm and add it onto tyrosine residues. Instead, what they do is they go to ATP molecules and they cut one of the phosphate groups off the ATP molecule, turning it to ADP, and they stick this phosphate group onto the alcohol groups of the tyrosine residues. Okay, but the principle is the same, that you're adding phosphate groups via phosphoester links onto tyrosine residues. Okay, right. So that's what this tyrosine kinase domain is capable of doing. It's capable of phosphorylating tyrosine residues. Okay, right. So what we now want to turn our attention to is how we activate these receptor tyrosine kinases. Okay, and they're going to be activated when the ligand binds to them. So at the moment, this tyrosine kinase domain that you have here, this enzyme that we have as part of the receptor, is active, but it's active at a very low level. So the actual activity of this is feebly low. Okay, so what's now going to happen is activation. So how does activation proceed then? So we're going to start off by binding ligands to our ligand binding domain of our receptor tyrosine kinase. Okay, so let's draw this picture again. Here we have our ligand binding domain, which I'll colour in orange here. Okay, so this is our ligand binding domain, and now a ligand molecule is going to come and bind to this ligand binding domain. Okay, so in turquoise, this little box that I'm drawing here is our ligand. Now, if we're thinking of epidermal growth factor receptor, then clearly this ligand will be the epidermal growth factor. Okay, but for other receptor tyrosine kinases, there will be other ligands. Okay, then we've got our membrane spanning alpha helix here, okay, which is straddling the membrane. And then on the cytoplasmic side, we then have our tyrosine kinase domain. Okay, now, what's going to happen then is once the ligand binds to the ligand binding domain of our receptor tyrosine kinase, you're going to get a change in conformation in the receptor tyrosine kinase. Specifically, you're going to get a heavy change in conformation in the ligand binding domain here. Okay? And this change in conformation is going to allow this ligand binding domain to dimerize with the ligand binding domain of another receptor tyrosine kinase of the same type. Okay, so if we're talking about the epidermal growth factor receptor, then we're going to get another epidermal growth factor receptor coming along, which also needs to have a ligand molecule bound to it. So this one can't bind to the first one unless it also has a ligand molecule bound to it, which in this case will be epidermal growth factor. Okay, so here's our ligand molecule. And once both of them have ligand molecules bound to them, they both undergo this change in conformation, and this means that they can now dimerize together to form a receptor tyrosine kinase dimer, like so. Okay, so here's the membrane spanning alpha helix, and here's our tyrosine kinase domain. Now, this brings these two tyrosine kinase domains extremely close together. Okay, and you remember I told you that the tyrosine kinase domains, although they're not particularly active at the moment, they do have some activity. They are still capable of catalyzing the addition of phosphate groups onto tyrosine residues. So what is now actually going to happen is a process known as trans-autophosphorylation. Okay? Now, trans-autophosphorylation is also sometimes it's more commonly just called autophosphorylation rather than trans-autophosphorylation, because autophosphorylation is a long enough word already without sticking a trans on the front, although trans makes slightly more sense than autophosphorylation. So firstly, let me describe to you what this is, and then I'll come back to why it is called this. Okay, right. So let's call this first receptor tyrosine kinase number 1. And let's call this second receptor tyrosine kinase number 2. Okay? And what's going to happen is that the tyrosine kinase domain of receptor tyrosine kinase number 1 is going to phosphorylate tyrosine residues on the tyrosine kinase domain of receptor tyrosine kinase number 2. Now, I will show the phosphorylation as just having this little blob, which I'll colour in pink here, stuck on. So that represents a phosphate group. So this is what tyrosine kinase domain of receptor tyrosine kinase number 1 has done to the tyrosine kinase domain of receptor tyrosine kinase number 2. Now, the tyrosine kinase domain of receptor tyrosine kinase number 2 is going to then reciprocate by sticking a phosphate group onto a tyrosine residue within the tyrosine kinase domain of receptor tyrosine kinase number 1. Okay, so now both of these tyrosine kinase domains have had phosphate groups stuck onto them. Now, what's the effect of this? Well, this is going to activate the tyrosine kinase enzymes. Okay, so their activity is going to go up, basically. So how can I draw this? I'll draw some sort of like sparks coming from it. Okay, so once we've stuck this phosphate group on, the tyrosine kinase domains are going to go from being extremely inactive to being extremely active. Okay, so they've been activated. Now what's going to happen is the tyrosine kinase domain on receptor tyrosine kinase number 1, which is now active, is going to phosphorylate a huge number of tyrosine residues that are in the polypeptide surrounding the tyrosine kinase domain of receptor tyrosine kinase number 2. Okay, so let me draw these here. So once it's now activated, it's going to turn its attention to tyrosine residues that are outside of the tyrosine kinase domain of number 2. It's going to phosphorylate other tyrosine residues around it. Okay, and similarly, of course, the tyrosine kinase domain of number 2 is going to phosphorylate tyrosines that are around the tyrosine kinase domain of receptor tyrosine kinase number 1. Okay, and there will be many of these. I've drawn 2, but there will be more than that. Okay, so there's many tyrosines within the cytoplasmic tail of each of the receptor tyrosine kinases that can then be phosphorylated. Okay, now this process is known as trans-autophosphorylation, or just autophosphorylation. So I'll start with why it's called autophosphorylation. So auto here. Auto means self, okay? So autonomy means self-governing, okay? So self-phosphorylation. Now the reason it's called self-phosphorylation is you view the dimer as being one structure now, okay? And the dimer is getting phosphorylated on tyrosine residues. And how is it getting phosphorylated? Oh, well, it's because it's got tyrosine kinase enzymes within it, which are phosphorylating it. So therefore it is self-phosphorylation. Trans-autophosphorylation kind of then acknowledges the opposite, that actually it's made up of two separate receptor tyrosine kinases, and what's actually happening is one is phosphorylating the other, and the other is phosphorylating that first one. Okay, trans means across, basically. So that kind of makes sense, because they're phosphorylating each other, basically. They're phosphorylating the one that is across from them. Okay, right. So both of those terms, it doesn't matter, they both mean the same thing. So what is now going to happen is the appearance of these phosphotyrosine residues that we now have on the cytoplasmic tail of both of the receptor tyrosine kinases in this receptor tyrosine kinase dimer. These phosphotyrosine residues are going to serve as docking sites for the next protein along, basically. Okay, and we'll see that next protein along in the next video.